Good morning, everyone. My name is Quexada. I'm a second-year PhD student at Université technologie de Compiègne and Universidad Nacional de Colombia, and today I'm going to present some of our work that explores the mechanisms of growth plate development and disease progression through a dynamic trabecular bone microstructure model. So, people usually think of the skeletal system as a rigid, non-dynamic structure, but the truth is that bone is highly active. It grows during the developmental stages and it continuously remodels throughout the human lifespan. Actually, the entire skeletal system remodels itself every 10 years. So think of that for a second. Each one of you is an entirely different person than it was 10 years ago, literally. It also serves as a mineral reservoir and gives structural support to our body. It does all these activities and more in function of mechanical and biological factors. So in this work, we focus on endoconal growth, that is the process by which long bones increase their length, and this happens in a region known as the growth plate, epiphyseal plate, or physis, shown here. And to study this, we focus on the effect of different mechanical factors such as stress and load frequency, and also biological factors such as the action of hormones and signaling pathways. And we look into how these correlate and affect growth development. For this, one of our major tools is mathematical modeling, which allow us to simulate, in this case, very specific scenarios, for example, how the growth plate presence may be affected by the trabecular patterns, and vice versa, how the trabecular patterns are influenced by the shape of the growth plate. We use micro-CT scans to validate the approach that we developed based on the works of Carter and Nakenhorst, and we used our model to study different diseases affecting the growth plate, such as leg calve breathless disease, Severus disease, sleep capital femoral head, and even hip dysplasia, which we use as a study case. And furthermore, we want to shed light on how the change in shape is determined by dosification bridges and the high shear stress zones in the vicinity of the growth plate. So I hope you find this work useful, and if you have any doubt, I will be happy to address it virtually or personally. Thank you very much.